morning. We are the breast biopsy device team. We are Savannah, Gabby, and Gwen.
1 in 8 women will develop breast cancer over the course of their lives. When a woman presents with an abnormal finding, a woman will often get a biopsy procedure to see if the tissue is cancerous. During this procedure, a core needle biopsy device is typically used, and a marker is typically used to mark where the location of the sample was taken from. Current devices in use are core needle breast biopsy devices, hologic marker clips, and vacuum-assisted biopsy devices.
Breast radiologists will use a time-efficient, easy-to-use, intuitive biopsy device that minimizes the number of insertions and reduces the procedure length to less than 30 minutes while collecting at least 80 milligrams of tissue for laboratory cancerous testing. Using this problem statement, we created a prototype that would combine what is currently used as 2 devices, being the biopsy needle and a marker needle, and we combined these into 1 device in order to limit the number of insertions and reduce the procedure length.
We have concluded, based on previous testings and user feedback, that these 3 needle iterations are the most promising. First, we made a dual collection needle that could collect tissue at the top and the bottom that, when scaled down to the original dimensions, can collect 1.85 times more tissue than the original device. However, users expressed concerns about the strength of the flat plate in between the 2 sections of needle. Next, we wanted to focus on incorporation of the marker device into the original biopsy needle. Our first iteration consisted of tubing and a pipe cleaner that would insert the marker into the target area. Our next iteration consisted of a 3D printed model of a SolidWorks design, in which at the end of the needle incorporated a hinge that would allow the marker to be placed into the target area. After a 100% success rate of inserting the marker into the target area and user feedback, we concluded that this design was the most promising to move forward as our final design.
From user feedback, we found that changing the handle design didn't help much in trying to limit the amount of time the procedure took. Therefore, we tried to iterate on the end of the needle, where our final prototype, as mentioned before, scaled up, will eject a marker into the patient's breast after taking a piece of tissue, which will eliminate about 1/5 of the procedure time, as the radiologist will not have to use a combination of 2 devices. First, the inner needle will eject. This will collect the patient's breast tissue. Then, the hinge flap will be opened by pressing the lever in the back end. The marker will come out, and the inner needle will slide back into the outer needle. To test the functionality of our device, we inserted it into jello, which mimics breast tissue, and tried to eject the marker 10 times. 100% of these trials were successful, and the marker did not falsely deploy. In future work, we need to work on scaling this device down to the appropriate size in order to test on humans, and we also need to attach the spring mechanism and combine the looks like and the works like model to be as one.
Following our key design inputs, our finalized prototype, as shown previously, meets all of them. The device is single-handed use. It collects at least 0.026 milliliters per insertion, with the ability to collect a total of 80 milligrams of tissue, as confirmed by previous jello insertion testing. Based on user feedback from Dr. Gershon, Dr. Stratton, and Dr. Goodman, the device is both comfortable and maneuverable, as well as innovative, as there is nothing currently on the market or patented that combines both a biopsy needle and a marker device. The market for core needle breast biopsies is expected to continue growing with a CAGR of 5.6% between 2016 and 2024. Our device could be used in all core needle breast biopsies where a marker is also placed in the breast following the removal of the tissue. The serviceable market is around 80 million, and with a conservative outlook expecting to get 5% of the total market, the service obtainable market is around 4 million. This device would save both time and money for our primary users and reduce time and pain for our tertiary users. The primary users would choose our device at $160 rather than buying a breast biopsy device and a marker device, which would total to around $230. And the patients would prefer our device as it limits the number of insertions, thus reducing the pain during the procedure. There are currently reimbursement codes for both a core needle biopsy and a marker placement, so the primary user would be able to receive reimbursement for both of these. The next steps would be to scale down the needle to the correct size and to patent our design. This would take about 2 years to patent and be ready for manufacturing and distribution.
The device classification is Class 2, meaning there needs to be a submission of a 510K. Previously, a predicate device is INRAD's Select Core Variable Throw Biopsy device, in which their 510K was approved November 17, 2009. This device also has a regulation number of 876.1075.
Thank you for listening to our video, and we are now open for any questions.